Hey everyone, welcome back to another episode of Genetics. Today we're going over the final section of Chapter 7, which is basically mRNA modification. And in this section, we're mostly focusing on eukaryotic organisms because eukaryotic organisms are the only ones that modify their pre-mRNA into mature mRNA. There are three ways how they do this. Firstly, at the 5' end, what's happening over here is that you're adding an inverted guanine to the 5' end. And this inverted guanine is called the 5' cap. So 5' cap. The enzyme that is responsible for this process is guanylyl transferase. And this will be the enzyme involved in adding the 5' cap. Additionally, a couple of nucleotides around the 5' cap, an enzyme called methyltransferase, will basically add methyl groups to this end, and that'll be the end of the 5' end modification. So yeah, methyltransferase adds methyl groups, 2 nucleotides around 5' cap. Around the 5' end, I should say. And then after we go over the next two, we're going to go over the significance of having these modifications, okay? Okay, so now we're going to go into splicing. So this happens in between these different ends. And if you're aware, every eukaryotic organism has an intron and an exon. So an intron and exons. The issue with these that a lot of students encounter is that they don't really remember which one is which. So one of these is a part that contains the genetic information for you to translate into a protein. And then the other part is just filler sequences that are going to get removed. But the issue is a lot of people don't understand which one is which. It can get a little confusing. So I'll just give you guys a quick little acronym for you to understand. So the introns stay in the nucleus to get degraded. And then exons exit the nucleus to get translated. So that's a quick way how you can memorize which one is which. Just remember introns for in the nucleus and then exons for exiting the nucleus. Okay? From this, the primary enzymes involved, or the primary proteins involved in this process are spliceosomes, which is a collection of mRNA, which is a collection of snRNA and other proteins that find these intron sequences and they splice them out. We don't have to necessarily know the exact process of this mechanism because this is, after all, a genetics class. But essentially what can happen here, you can have different kinds of splice variants because you can have different kinds of spliceosomes. So just remember that this process can lead to you having different splice variants. which are called this process is called alternative splicing. So just remember that. Additionally, one of the things that can result from splicing, you can have mutations, which we're going to cover a little later on in the class. And then that's basically it for splicing. Just remember that introns stay in the nucleus and exons exit the nucleus. And they're the result of these spliceosomes, which is a collection of proteins and snRNA that work together to find the correct sequence that you want to get rid of. Okay? And then at the 3' end, the last modification is called it's being done by a poly-A polymerase. So this poly-A polymerase, as the name suggests, you basically add a ton of adenines. So you add a ton of adenines to the 3' end, and then that'll be your final modification that you need to have. Okay? Now the significance of you doing these modifications, like I said, the splicing significance is that because you have splicing, you can end up with different kinds of proteins from the exact same mRNA molecule. So you can have different types of variants from the same mRNA molecule. So that increases the amount of variation, which is always good for a biological organism to have increased variation, especially with its proteins. The 5' end and 3' ends are primarily involved for nucleus transportation, so being transported out of the nucleus and preventing the mRNA from being degraded by nucleases. So by adding that 5' cap and by altering the length of the adenines on your 3' end, you can change the sensitivity to the harmful substances that you can find in the cytosol for it to be degraded by a nuclease or something like that. So basically that's pretty much it. There really isn't that much into it. Just understand the three changes, and that's it. Have a good one.